Hello, biology, and welcome to chapter 11, which is all about gene expression. In the last chapter, we talked about DNA synthesis, we talked about transcription of mRNA, and we talked about the translation of that mRNA into protein. And this is how gene expression happens. We take something from the DNA, and we turn it into mRNA, and we take it to protein. So when we talk about phenotypes, we're talking about protein structures that cause function or that cause different kind of traits. For me, having a brownish blonde hair and having a red beard is my phenotype. It is caused by the proteins in the hair molecules that I create and how they reflect light and how they look to the human eye. So let's jump into gene expression, and we'll talk about what it is, how it happens in bacteria, and how it happens in us humans. All right, so the role of gene expression. Gene expression is the turning on or turning off of a gene that results in transcription of that gene and produces mRNA. And then that mRNA, most of it is going to go and be translated into protein. And the reason that we need this is you don't always need the genes for every cell all the time. We don't need adrenaline in our tongue cells all the time. So we only make it when we need it. All right, so recap, some real quick protein function. Remember that lots of proteins are used for structure, things like hair molecules, things like fingernails, things like cytoskeleton. But a lot of proteins have the job of being catalysts. And catalysts, remember, are these enzymes that aren't used up that help facilitate kind of chemical reactions. This one here is hemoglobin. Hemoglobin is essential for picking up oxygen from the lungs and taking it to the cells, releasing that oxygen to the cells and picking up carbon dioxide, taking that to the lungs, and then expelling that as waste. So we don't produce hemoglobin in all of our cells. We only produce hemoglobin in cells where it's needed. Most of the time, that's in blood cells, and then there's a myoglobin, which is a muscle variety of hemoglobin that is used in the muscles to help move kind of oxygen around. Now, it's important to bring back this term of genome, the complete genetic material contained in an individual. You only can express genes that you currently have. So by regulating gene expression, you can turn the genes that you currently have either on or off. You don't express all of them all the time. Some of them you only express in your childhood, and then by the time you're an adult, you've stopped expressing them, and you no longer express those genes. These are why we stop growing, where our growth plates solidify. Instead of just growing our entire lifetime, those genes shut off, those proteins that kind of help and push that growth process stop being produced and stop having that effect. Real quick, we got to have a recap on prokaryotes before we talk about the DNA. Remember that prokaryotic DNA exists in one circular chromosome, and that compared to eukaryotes, prokaryotes are very simple, and that they divide using a process called binary fission. This is kind of important as we go into. We're going to talk about prokaryotes first and how they use gene expression, and then we'll talk about eukaryotes. So, gene expression in prokaryotes. The two guys to remember here who did a lot of work are Francois Jacob and Jacques Monod, and they discovered how genes control metabolism in E. coli, Escherichia coli. E. coli lives in the guts of mammals. You guys have it. It's very important for your synthesis of vitamin K. But E. coli has the ability to use this molecule, lactose, for energy. Now, most of the time, we kind of think of glucose as energy, and this is a molecule of glucose right here. But lactose is a molecule commonly found in milk, and it has this galactose unit here and this glucose unit bonded. E. coli are able to use this for energy, but only under certain circumstances. So when E. coli is in an environment that has lots of glucose, it uses cellular respiration, just like us, a little bit different because they don't have mitochondria. But when it enters an environment that has lots of lactose and no glucose, it activates genes to now utilize that lactose. So it's able to switch which sugar it's using for energy. And the gene for this is coded for by 3 enzymes. And these 3 enzymes are very important to each one of them is kind of a sequential breakdown of lactose into usable products. But let's talk about the regulation of this enzyme production. So when we talk about genes, we have several kinds. We have structural genes, and these are genes that code for polypeptides. We have a promoter, which is here, in this little P area. This is a DNA segment that is recognized by the enzyme RNA polymerase. RNA polymerase is very much like DNA polymerase, in that it makes RNA molecules one molecule at a time. And then we have an operator, and this is a switch that allows RNA polymerase the ability to bind to this promoter and start making what we call this operon. And an operon is a series of genes that code for a specific product and the regulatory elements that control these genes. So the whole gene here is the operon. The actual ones that make polypeptides are the structural genes, and the control genes, the promoter and the operator, are their own kind of thing. But the whole thing is the operon. These guys are usually named after what they do. So the one that makes lactose in E. coli is called the lac operon, kind of a shortened version of lactose and then just operon. So with operon control, like we said, these can be turned on or off. E. coli does not constantly use lactose. When they're using lactose and they're placed back into a glucose-rich environment, they start using glucose again, and they turn off that lactose gene, and they no longer use it. So it's very circumstantial which genes a cell will use. So when the operon is turned off, this is because lactose is absent. So what happens during this is a repressor protein is going to attach to the operator. And the repressor protein is any protein that inhibits genes from being expressed. And it's got its own set of genes called regulator genes. These are specifically genes that code for repressor proteins. So everything's got something that codes for it in the DNA. So how do they stop these gene expression here? Well, repressor genes here, they don't allow this RNA polymerase they try to attach to the promoter here, but they can't because this repressor is blocking it. So it's unable to slide down this molecule. In this case, we're trying to make the amino acid tryptophan, but they're unable to slide down this operator. So this repressor is stopping this RNA polymerase from making RNA and taking that signal. When this is turned on, usually what will happen is something will bond to this. In this case, this tryptophan bonds to the repressor and releases it off of here. And now the repressor is able to slide down and make all those genes. In the lac operon, lactose has to be present. It bonds to the repressor and it says, hey, there's a ton of lactose in here. Can you not be here so I can do this RNA polymerase and just make all this? And that's kind of how that works. All right, inducer. These are molecules that initiate gene expression. So lactose binds to the repressor protein, and it induces that repressor protein, induces that operon by removing that repressor protein. So without lactose, this whole thing is turned off. But when it runs, this DNA polymerase will come over here. It will make these lac operon mRNAs over and over and over again. That will make these genes to use lactose. But the reason that we have inducers is because it allows that cell to only use one gene at a time. Lactose, E. coli is only going to use either cellular respiration or it's going to use this lactose kind of metabolism. It's not going to do both of them. It's going to choose one or the other. All right, now we're going to jump and we're going to go to eukaryotes. They're a little bit more complex. Eukaryotes are much larger. They have larger genomes. They have many different chromosomes instead of just one circular chromosome. And the same thing, only certain amount of genes are expressed at any given time. However, unlike bacteria, we don't have operons as commonly. We have kind of more complex genomes. So let's talk about the structure of a eukaryotic gene. Remember that DNA is always going to be organized as fibers of chromatin wrapped around proteins called histones. This is when DNA is very densely packed in chromosomes. After mitosis, the DNA uncoils back into euchromatin, and you can't transcribe and make genes out of the DNA unless it's in the euchromatin phase. So euchromatin is this loose storage of DNA here, where it's not this heterochromatin, which is very densely packed. When it's like this, the DNA and RNA polymerase can get in there and actually get to work. So transcription can only happen when it's euchromatin. Some portions of this, especially like around the histones here, never get uncoiled, so their genes are never transcribed. They're never used for any kind of genetic control. So there are genes that we don't even use inside of the human body. All right, the gene structure is a little bit different. Like operons, they will have promoters, and this is where RNA polymerase will bind. And then inside of a DNA segment of a eukaryotic gene, there are two kinds of segments. There's introns and exons. And introns are segments of DNA that are transcribed but not expressed, and exons are segments of DNA that are expressed. So when we cut this whole gene out here, this whole thing here, these blue bits are not important for this gene. Let's say it's eye color, and these parts just are not in that gene. Because our DNA is a little bit more complex, we use the same segments of DNA over and over again for different genes, but depends on what we take in and out of them. What happens to these introns after we transcribe them? So after we turn it into mRNA, the introns are snipped out, and the exons are then stitched together to form a gene. So what that means is that different genes use different introns for the same promoters. The same segment of DNA has chances to be many different proteins. Most of eukaryotic control is after transcription. So in prokaryotes, transcription occurs in the cytoplasm. In eukaryotes, this is in the nucleus. And then for both, we translate in the cytoplasm. So after transcription, we have both introns and exons. This forms pre-mRNA, which is a form of messenger RNA that contains both introns and exons. So if you see both important and non-important chunks of RNA in here, that is pre-mRNA. When we splice introns, we'll take these introns out, we'll splice these exons together, and then we'll make this mature mRNA that then leaves the nucleus and goes about into the cell to find a ribosome. So, control at the onset. Most of our regulation is going to occur when RNA polymerase binds to a gene. These regulatory proteins in eukaryotes, we call them transcription factors. Transcription factors are very important for whether or not you're expressing a gene, using a gene, making a hormone, not making a hormone, etc., etc. We have these things in eukaryotes called enhancers, and these are transcription factors that bind to sections of DNA that are far away from genes. And what they do is they end up taking the DNA that's in this big long cord and they scrunch it together so that these two chunks are close together, so that we can translate it much quicker by having this DNA closer together. All right, 11.2 is all about gene expression in development. So it's this taking the same thing here and then talking about from zygote up to an individual. And it's important to note that all multicellular sexually reproducing organisms begin life as a fertilized egg called a zygote. And you can see here a zygote has the DNA from dad, who is the sperm, and mom, who does not, has both of those DNAs. And the zygote is very cool because at this point in time, this one cell has the entire genome, every single gene needed to make an entire organism. As a human, you started out as one cell, and now you are millions, if not billions, of cells that are now working together that started from this one single cell. As cells get older, they go through this process called cell differentiation. where development of cells to have different functions happens. Cells will turn into different kinds of cells: immune cells, epithelial cells, nervous cells, blood cells. These cells will form tissues. When we talk about bone, bone is a tissue, or you can call bone an organ. Just kind of depends on your definition of it. But if we take a muscle and we form it in muscle cells and we form it into a whole bunch of them, we have muscle tissue. And then if we form a whole bunch of those tissues together, now we have a bicep, which is an organ. We have a muscle organ now. An important class of genes are the homeotic genes. These are regulatory genes that determine whether certain anatomical structures develop. So this is like why you have arms and legs and why you only have 2 of each. Or normal humans, we'll say normal humans have 2 of each. So homeotic determines what appendages develop, when they develop, where they develop. Make sure that your eyeballs are in the right spot, your nose is in the right spot, you have 2 ears instead of 4. Because of their importance, we call these ones master genes. And these guys, like any other gene, get transcribed and they get translated. When this happens, we get regulatory proteins. These proteins turn on or turn off certain developmental genes to make sure that you as an organism are developing, and then you stop developing. You don't want 3 arms, so you stop developing after you get 2. The best example of the homeotic genes that we can talk about is the HOMEOBOX sequence. And the HOMEOBOX sequence here is 180 nucleotide sequences that all genes share. And in young animals, the HOMEOBOX determines developmental patterns. In adults, that changes and it turns into more regulatory. It turns into more like maturity and puberty and things like that. So it's it changes a little bit, but you can see that this protein here has this homeotic gene, this purple gene inside of it. Now, usually it will get cut out and it won't be used, but it's in all of these genes because it's very important. All right, so now we're going to talk about gene expression, cell division, and cancer. So the division of cells is regulated by many genes. Remember in mitosis, we talked about the different checkpoints. At each of those checkpoints, these proto-oncogenes, which are genes that regulate cell growth, cell division, and the ability of cells to adhere to each other, are making sure that these cells are undergoing proper mitosis, getting the right amount of DNA with no errors, right amount of organelles, right amount of cytoplasm, all of that's regulated. So the proto-oncogenes code for proteins that regulate cell division to happen at the correct sequence and the correct rate. They will need to happen not too fast, not too slow. What can happen to these proto-oncogenes is they can turn into oncogenes. When a mutation changes these proto-oncogenes, they turn into oncogenes. These genes can cause uncontrolled cell proliferation. And what that means is either there's overstimulation of cell division or there's uncontrolled cellular division. So now these cells are not going through this regulation. They're just dividing really, really quickly without checking their DNA, without checking their size, without checking for organelles. All of those important steps we need to do to make sure we get healthy cells. When this happens, we get development of tumors. And tumor is any kind of abnormal proliferation of cells. Here you can see some benign tumors on this guy here. These are all resulting from uncontrolled growth. So even though these benign tumors pose no threat to life, they will still continue to grow unless they're removed. So even though you have a benign tumor, it's still good to get that thing removed. They can grow large enough to compress vital organs. This is really important in breast cancer, where you got a lot of important stuff, the heart and the lungs and the esophagus all right together, right around the mammary tissue there. Skin cancer, most common kind of benign cancer, and these usually can be removed with surgery, and usually should be. They're usually pretty painless. You're in one day and you leave the same day, and you don't have any kind of hospitalization. Now, the thing we worry about are things called malignant tumors. And these are uncontrolled dividing cells that invade and destroy healthy tissue. They're wanting to go other places and they're wanting to cause damage. This uncontrolled growth invades, destroys healthy tissue elsewhere in the body, and this is when we start to call it cancer. Now, normally your body has what we call tumor suppressor genes. These are ones that proteins that prevent cell division from occurring too often. They kind of put a waiting period on these cells to say, hey, you can't just keep making more of yourselves. You've got to grow up, you've got to do your job, and then you can make more of yourselves. But if this gets damaged, again, this cell here, now all of a sudden we have these cancerous cells that can form. So when one is kind of broken, the cell is still kind of in control, but not, it's losing it. And then usually after one type of tumor suppressor gene gets mutated, others follow quickly. And so after several kind of mutations here, we now have a malignant cell versus these healthy cells that have none of these regulatory elements anymore, and now they are able to spread and cause kind of damage to the rest of the body. So genes are expressed in cancer. Normally, your cells are attached to other cells by a membrane, and normally mammalian cells, like human cells, stop dividing after 40 cell divisions. In metastasis, this is when we get the spread of cancer cells beyond their original site. So here we have this original tumor here. It gets into the bloodstream and spreads to a secondary kind of area. Now, also with cancer cells is they can divide much more than 40 times, and they do not have to be attached to other cells. So they will travel throughout the body to other cells and start to cause cancer there. So some causes of cancer. It can occur randomly. It is an unfortunate part of human biology that we have certain genes that can just randomly mutate into cancer. But most oftenly, they're due to what we call a carcinogen. And carcinogen is any substance that can induce or promote cancer. So when you see those little warnings on labels, do not use if you're pregnant, it may cause cancer, that's what they're warning you against, that this thing is a carcinogen. Most carcinogens are also mutagens, and mutagens are agents that cause mutations to occur within the cell. And we're going to talk about that more in a further chapter. Some well-known carcinogens, if you're in the workplace, this signal here means that whatever you're dealing with, whatever liquid or dust or gas, is a known carcinogen, and that inhaling, ingesting, coming in contact with it has the ability to potentially mutate your DNA and cause cancer. But ones that we know are tobacco smoke, ultraviolet light from the sun. This is where you always wear sunscreen. Always wear sunscreen. If you learn one thing, do that. It's important. Some other things that can cause cancer are viruses. Viruses promote replication of cells because they need to use the cell's machinery to make more viruses. More cells that have the virus means more virus can get made. So sometimes they turn off these tumor suppressor genes to make more virus, and in turn give the individual cancer. Just briefly, kinds of cancer. There are lots of them. We're just going to talk about a couple. Carcinomas. These can grow in the skin and organ tissue. Things like lung and breast cancer are usually carcinoma. You'll have lots of things going on there. You can have sarcomas, which are tumors that grow in the bone, or this one is in muscle tissue. It's like peeking through there. This is a sarcoma. And then lymphomas are solid tumors in the lymph system. And when you get sick, your lymph nodes under here, usually in the back of your throat, will swell up. You can have them on your knees or your elbows, or it's not elbows, your armpits. But when you get lymphomas, you actually get solid tumors in these that kind of block the system and reduce your ability to have an immune response. The last one we're going to talk about is leukemia, and this is a tumor of blood-forming tissue. And this is when we get white blood cells uncontrollably forming here. And it usually takes years to develop and to form, and it's pretty serious usually. It's a lot of blood transfusions. Normal blood has a lot of these red blood cells, these little guys. Leukemic blood, you can see the white blood cells are taking over, and with that, they don't carry oxygen as well. They don't get rid of carbon dioxide. They don't carry sugar as well. So these guys are no bueno. These guys are bueno. So that is the conclusion to chapter 11. I hope you learned something new. If you have any questions, let me know, and we'll see you in the next one.